The research interest in my laboratory is to investigate the mechanisms that promote vascular damage during obesity. Obesity is a growing epidemic that disproportionately affects Blacks and Hispanics, and obesity increases the risk of cardiovascular disease pathology. This is due to vascular remodeling that occurs as a result of abnormal function of smooth muscle cells and endothelial cells. In my laboratory, we found that SGK1 is upregulated during obesity, and others have shown that obesity leads to mitochondrial dysfunction. So we hypothesized that metabolic reprogramming by SGK1 contributes to abnormal vascular smooth muscle cell function and subsequent vascular damage during obesity. Our goal was to investigate a role for SGK1 and a novel mediator that we identified, the mitochondrial calcium uniporter, in regulation of obesity-mediated metabolic reprogramming. Our work takes advantage of a smooth muscle cell-specific knockout mouse that was developed in our laboratory. We used these mouse in a diet-induced model of obesity, harvested the aorta and assessed vascular remodeling, and then extracted smooth muscle cells from the aorta and analyzed cellular respiration, specifically glycolysis and oxidative phosphorylation, using the seahorse technology. We also measured gene and protein expression of SGK1 and MCU. The data is shown here. We show that obesity induces upregulation of SGK1 in vascular smooth muscle cells. We also show that obesity leads to an increase in vascular remodeling in wild-type animals, and that knocking out SGK1 specifically in smooth muscle cells abolishes obesity-related vascular remodeling. We also show that vascular protection correlates with vascular smooth muscle cell metabolic reprogramming, which is due to an increase in oxidative phosphorylation. When examining the downstream mediators that may mediate this effect, we find that the mitochondrial calcium uniporter is enriched in vascular smooth muscle cells that are devoid of SGK1 when these cells have been exposed to a high-fat environment. Knocking down or inhibiting MCU with the potent inhibitor RU360 leads to a decrease in oxidative phosphorylation, indicating that metabolic reprogramming is due to MCU-dependent OXPHOS stimulation. So we've identified a novel mechanism showing elevation of SGK1 in vascular smooth muscle cells during obesity negatively regulates mitochondrial calcium uniporter, contributing to metabolic dysfunction and vascular remodeling. And we identify SGK1 as a potential novel therapeutic target for obesity-related vascular diseases. Thank you.